Hello everyone, in this class we'll try to learn about hemoglobin. Heme portion of hemoglobin is synthesized from glycine and succinyl-CoA. Heme is synthesized by cells of erythroid series in red bone marrow. Hemoglobin starts appearing in the developing of RBC at the intermediate normoblastic stage. These are the different steps of synthesis of hemoglobin. 2-succinyl-CoA and 2-glycine forms the pyrrole ring. 4 pyrroles, they are going to form protoporphyrin ring, and protoporphyrin with iron, it is going to lead to formation of heme. Heme and polypeptide, depending on the alpha or beta chain, they're going to form hemoglobin chain, and 2 alpha chains and 2 beta chains together, they will form hemoglobin A. Hemoglobin is a globular structure made up of 4 subunits. Each subunit contains heme moiety conjugated to a polypeptide which is called as globin. Heme is an iron containing porphyrin derivative. There are 2 pairs of polypeptides in each hemoglobin molecule. Hemoglobin A consists of 2 types of polypeptides, that is alpha and beta, and one hemoglobin molecule can combine with 4 molecules of oxygen or 8 atoms of oxygen. So this is the structure of hemoglobin, which is made up of the heme structure. So this is the structure of the heme, and these are the 4 polypeptides associated with each hemoglobin. In each hemoglobin, you have 2 chains, 2 alpha chains and 2 beta chains. Normal values: males have 14 to 18 grams percent of hemoglobin content in their blood, whereas females little less, averages around 14 gram percent. There can be different types of hemoglobin. The adult hemoglobin, there are 2 types, hemoglobin A and hemoglobin A2, where the hemoglobin A has alpha 2 and beta chains, whereas hemoglobin A2 has alpha 2 and delta 2 chains. Whereas fetal hemoglobin has alpha 2 and gamma 2 beta chains, and functionally fetal hemoglobin is different from adult hemoglobin, that it has a more affinity towards the oxygen. Some of the abnormal varieties are seen in hemoglobinopathies, and these are hemoglobin S. In the hemoglobin S, in the beta chain, glutamine at the 6th position is replaced by valine. When HbS is reduced, it precipitates into crystals within the RBC, leading to sickling of RBCs, and it occurs whenever the RBCs are exposed to the hypoxia. And sickle shaped RBCs are more fragile, resulting in sickle cell anemia. The other hemoglobinopathies are hemoglobin C, hemoglobin E, and hemoglobin I. Thalassemias. Both alpha and beta chains are present in these diseases and are normal in structure, but produced in reduced amounts, or sometimes they are absent. There are variants in thalassemia. It can be major beta thalassemia or minor beta thalassemia. Major beta thalassemia is also called as Cooley's anemia or Mediterranean anemia. It's less common. There will be total absence of chain synthesis. It has a homozygous transmission, and victim usually dies because of the major beta thalassemia, because of excessive of hemolysis. Minor variant is more common, and it occurs because of the partial synthesis of beta chain, and it transmitted heterozygous pattern. So these are the variants of thalassemia. It can be beta thalassemia because of the absence or reduced beta chain synthesis, or it can be because of the reduced alpha chain synthesis, which is rare. Beta thalassemia can be major and minor. Minor is more common because of it occurs because of the partial production of the beta chains. Heme-heme interaction. In the initial phase of oxygenation, the combination of heme and oxygen is bit slow, but once a little of oxygen has combined with the heme, further interactions are facilitated. This is called as heme interaction, and this explains the sigmoid shape of the oxygen dissociation curve. In the oxygen dissociation curve, initially the uh, the association of oxygen with hemoglobin is linear, but at particular point of time, the combination with oxygen hemoglobin, it increases tremendously. So that is why it leads to the sigmoid shaped oxygen dissociation curve or association curve. Coming to the catabolism of hemoglobin, when RBCs become old and their walls become weak and their shape changes, and finally RBCs are broken down. RBCs are destroyed mainly in the spleen by the reticuloendothelial cells. Hemoglobin is then released from RBC, which splits into heme and globin. The end products of heme catabolism are bile pigments. Bilirubin has no function in the body, so excreted through the bile. Globin is broken into amino acids within the reticuloendothelial cells, which will be later utilized for synthesis of body proteins. Iron is then released into the plasma and is transported by transferrin to the storage sites. Porphyrin ring is broken down in reticuloendothelial cells of liver, spleen, and bone marrow, where it gets converted into biliverdin, that is a green pigment. Biliverdin gets reduced to form bilirubin, which is red orange in color, which is then released into the plasma. This unconjugated bilirubin, soluble in lipid solvent, is carried by plasma in combination with albumin to the liver, and it is this form of bilirubin is not excreted in the urine by kidney. In the liver, bilirubin undergoes conjugation with glucuronic acid and forms bilirubin monoion diglucuronide, which is soluble in water. Bilirubin diglucuronide is hydrolyzed and reduced by bacteria in the gut to urobilinogen, which is colorless. Most of the urobilinogen is oxidized by intestinal bacteria to stercobilin, which gives feces the brown color. Some of the urobilinogen enters the portal blood. The remaining urobilinogen is transported by the blood to the kidney, where 5% of it is converted into yellow urobilin and excreted, giving the urine its color. Most of the reabsorbed urobilinogen is re-excreted by the liver back to the gut. So this chart shows the catabolism of the RBCs. Okay, so when they're fragile, they get destroyed in the reticuloendothelial system, where they're going to release the free bilirubin, which is bound later in the plasma to the albumin. So this free bilirubin, that is unconjugated bilirubin, is taken up by the liver cells, where it gets conjugated, and this conjugated bilirubin is excreted in the GI tract and in the intestine. By the bacterial action, they get converted into urobilinogen, which is colorless, and because of the oxidation, they get converted into uh, stercobilinogen or stercobilin, and they give characteristic stool color. And some of the urobilinogen, uh, it is uh, it is brought to the kidneys, and where some amount of the urobilinogen gets excreted, and upon oxidation it gets converted into urobilinogen, which is important, which gives a characteristic yellow color for the urine. So this is in brief about the hemoglobin and its metabolism. Thank you.